We have just completed a reasonably extensive survey of bacterial and viral pathogenic species, and with that we should be starting to get a more comprehensive picture of infectious diseases as a whole. But now it's time to switch gears and dig into some more general microbiology concepts so that we can get a broader understanding of this topic, and we are going to start by examining the indispensable tool of the microbiologist, the microscope. As we learned at the very beginning of this series, microbiology is the study of microscopic organisms, and to study this otherwise invisible world, microbiologists use microscopes. This tool allows scientists to observe microscopic tissue and organisms with magnification ranging from 1200 to 1 million times larger than its original size. But how exactly did we get from examining a specimen simply by pulling it very close to our faces to being able to look at a screen of a sample enlarged enough to study the details of proteins within a single cell? Some of this information was outlined in the introduction to this series, but let's get a more sophisticated version of that story now. To begin this tale, we have to go back in time a few millennia. The very first signs of crude magnification tools appeared in about 710 BCE with the Nimrud lens, which was no more than a crystal that people would use as a magnifying glass. This was found in Nimrud, now modern-day Iraq. Later on in history, reading stones and eyeglasses were also used to enhance the eyesight of individuals. Now let's fast forward to 1595 CE, when the invention of the first compound microscope, or microscope with two or more lenses, was invented. The invention was attributed to Zacharias Janssen, although the records show that he would have been in his teens at the time, so there is speculation that his father, Hans Janssen, helped him with this invention, since they both worked as spectacle makers in Holland. Before this microscope, the smallest object people could visualize was the width of a human hair, which might seem small, but is actually quite large compared to the microbial world. Janssen's microscope allowed for the visualization of objects 9 times their original size. Early advances in microscopy after the invention of the first compound microscope revolved around the aim of producing better resolution, as improving magnification was not as tricky. Resolution is the ability to distinguish between two objects as separate entities, as opposed to the objects appearing smudged together. For example, currently the best resolution of a light microscope is 0.25 micrometers, which means that two objects can be a quarter of a micrometer away from each other without blurring together. As a point of reference, 1,000 micrometers make up 1 millimeter, which are the smallest lines on a standard ruler. After the invention of the compound microscope, the next big development in microscopy was brought about by a Dutch merchant, Antony van Leeuwenhoek, who made the first microscope that could visualize an individual cell. Leeuwenhoek made over 500 prototypes to get to the final version of his microscope, which could visualize objects 200 times their original size, a significant improvement from Janssen's microscope. This development led to Leeuwenhoek's discovery of bacteria, protists, sperm cells, and much more, as we discussed when we referenced his animalcules in the introduction to this series. At this point in time, the world of microorganisms was unlocked and available for study, but the look and accessories of the compound microscope were still far from what we recognize as a microscope today. It wasn't until 1665 that Robert Hooke, a scientist from the United Kingdom, added features to the microscope like the stage to hold the specimen, the illuminator to shine light on the specimen, as well as focus controls. These accessories allowed scientists to have more control over how they view the specimen. Additionally, these accessories made using the microscope much simpler, such that individuals would require very little training to operate it. The major innovations that were to follow didn't arrive until the 20th century, but each was truly significant. First, there was the evolution of the microscope from light or optic microscope to the phase contrast microscope in 1930. Then, the transmission electron microscope in 1931, followed by the scanning electron microscope in 1942. Finally, in the latter half of the century came the confocal microscope in 1978 and the scanning tunneling microscope in 1981. Advancements in microscopy are still being made to this day, so there is no telling what is around the corner. But over the next few tutorials, we are going to go into more detail regarding the functions and applications of some of the types we just mentioned, which remain the most used techniques in microscopy today. So without any delay, let's move forward and learn more about microscopy. Thanks for watching, guys. Subscribe to my channel for more tutorials. Support me on Patreon so I can keep making content. And as